Good afternoon, everyone, and thanks for joining the summit. Today, I'll be talking about my postdoc work, understanding the immunosuppressive functions of cancer-associated fibroblasts, CAFS, in lung cancer. Before we dive into this exciting project, I'd like to introduce myself. I'm Handan. I did my postdoc at Merck in discovery oncology and was fortunate enough to transition into a senior scientist and continue working for this great company. Prior to working at Merck, I did my PhD in the Department of Pharmacology and Cancer Biology at Duke University. Before further ado, let's get started. As we know, the tumor microenvironment is composed of all sorts of cell types, including tumor cells, endothelial cells, immune infiltrates, as well as cancer-associated fibroblasts. Now we have so many different therapeutic interventions that have been approved for the use of cancer treatment, such as chemotherapy, anti-angiogenesis therapy, as well as immunotherapy. However, one of the cell population that hasn't been successfully targeted so far is cancer-associated fibroblasts, CAFS. So the reason why we think understand CAF biology is important is that from several clinical trials, we found that stroma cells may mediate the resistance to immune checkpoint blockade. So here is the seminal work from our colleagues, and you can see the graph over here is that the y-axis is the tumor mutational burden, and the x-axis are the T-cell inflamed gene expression profile, GEP, and you can clearly see that those, and we found that those non-responders to pembro are localized in those patients with low mutational burden, as well as low T-cell inflamed gene signature. And more interesting is that the gene signature enriched in those patients are related to stroma biology and CAFS. So what are the known immunosuppressive features of CAFS? Let's first look at the confocal image of a human colorectal tumor section. The EpCAM marks up the epithelial cells, and the FAB marks up the cancer-associated fibroblasts, and the CD45 are the tumor-infiltrating immune cells. You can clearly see that the CD45 positive immune cells are largely restricted in this stroma cells, the FAB positive fibroblast portion. So if they want to get to the tumor bed, they have to swim across a sea full of fibroblasts. So consistent with our finding, there are also other studies show that CAFs may function as physical barriers to prevent the infiltration of immune cells into tumor bed. There are also other mechanisms. One of them can be that the interaction between the CD45 cells as well as the CAFs because of the close proximity. So one of the theory is that the CAFs can help to promote the generation and recruitment of immunosuppressive cells to sort of build up the immunosuppressive tumor microenvironment. The other mechanism can be that CAFs themselves can produce immunosuppressive molecules to dampen the anti-tumor immune responses. Here we are especially interested in the interaction between CAFs and tumor-infiltrating immune cells. So first, we like to unbiasedly understand how CAFs regulates tumor-infiltrating immune cells. We leveraged the publicly available TCGA datasets to see if there are any association. We decided to mainly focus on non-small cell lung cancer because those patients only partially respond to immune checkpoint blockade. Interestingly, we examined the gene expression of FAB. So as I mentioned earlier, it's a marker for CAFs, as well as several immune cell markers such as CD14, CD8, CD20, as well as other markers. We found that FAB is only strongly correlated with monocytic myeloid cells, such as CD14, CD16A, CD33. So we next want to validate this positive correlation in a separate cohort of primary lung squamous cell carcinoma using imaging mass cytometry. As we know, this technique allows us for multiplex detection of more than over like 35 protein markers. How we perform regular immunofluorescence staining also applies for IMC. The only difference here is that instead of using fluorophore conjugated antibodies, we use metal conjugated ones. The slides were then scanned by mass spectrometer, and here we're using Hyperon imaging system. And the spatial distribution of markers of interest is visualized by intensity of corresponding conjugated metal. We first did H&E and P63 staining to confirm the samples we use here are lung squamous cell carcinoma. Then we stained the samples with 17 IMC antibodies, including markers for fibroblasts such as FAB, alpha-SMA, CD19, and collagen, as well as several immune cell markers as shown here. So clearly you can see that in donor 1, we see more FAB positive fibroblasts compared to donor 2, and we'll also see higher percentage of CD14 positive cells in donor 1 compared to donor 2. However, how can we translate this information from images to actual numbers and graphs so that we can also analyze the positive correlation relationship? Luckily, our site-off core manager helped us to connect with Dr. Denise Scaparo from Harvard Medical School. He kindly showed us how to use the established MC data analysis pipeline by using those all publicly available software. And also he is the inventor for this wonderful software called HistoCat. Basically, we're able to extract protein expression at a single cell level and import the information onto HistoCat to visualize and quantify marker-specific cell types, as well as determine cell-cell interaction. So here are the example graphs of two-dimensional graph TC plot from 10 patients. So we're able to group similar cells together, and the warmer the color indicates the higher expression of specific markers. So you can see here those are E-coherent positive likely tumor cells, and those are CD20 positive B cells, and those are the CD8 positive T cells, etc. And we're also able to break down those testing plots into individual donors, so that we can able to get out those CD4 positive or CD14 positive cells and quantify the percentage of the cells in each of the patient. By doing this, we're able to confirm what we found in our genomic data analysis, and we found that the percentage of FAB positive cells is indeed positively correlated with monocytic myeloid cell populations by utilizing the IMC technique. So by leveraging HistoCat, there are other questions we can ask, one of which is whether these two cell types, CAFS and monocytic myeloid cells, can meet up with each other in the TME. In other words, whether they are neighbors, so that CAFS can further influence the phenotypes of tumor infiltrating monocytic myeloid cells. By doing this, we can perform an unbiased and systematic neighborhood analysis by using a permutation test, which is built inside in histocat to compare the number of interactions between all cell types in a given image to that of a matched control containing randomized cell types. So for example, if we want to understand whether the red cells are significantly interact with each other, we can first ask histocat to count the cell-cell interaction between the red cells in the matched randomized controls, or we can also count the cell-cell interaction in the given image from your tissue sample of interest. And then we can run permutation tests to see whether those interactions are significant or not. By doing that, the first thing we need to do is to figure out the cell cluster based on protein expression. So from the heat map, you can clearly see that cluster 4 are likely to be monocytic myeloid cells because it contains several monocytic myeloid cell markers such as CD14, CD33, and CD16. And cluster 5, 6, 12 are likely to be CAFS subclusters because those cells express CAF markers such as Fab, collagen 1, and CD90. And well, now let's see if those cell types are neighbored or avoided. So the red color indicates interaction, and the blue color indicates avoidance. And each of the highlighted squares indicating percentage of cells with significant cell-cell interaction. So I'm highlighting here, you can see cluster 4 did significantly interact with cluster 5, 6, and 12. And you can also correlate to what's the percentage of images here that showed positive correlation. To functionally characterize the association between CAFS and monocytic myeloid cells, we isolated CAFS from 3 primary fresh LSCC samples. The cells were confirmed by expression of Fibroblasts markers such as Fab, CD90, and PDGF-beta by flow cytometry. And indeed, these markers you can see here were not detected in long-squamous carcinoma tumor cell lines. We sought to identify soluble factors produced by the tumor-derived CAFS, and we found that all 3 CAFS consistently secreted high levels of monocyte and neutrophil attracting chemokines, including CCL2, CCL7, CCL1, 5, and 8. And we also found that those CAFs secrete MINFIL6 and VEGF. Those has been reported to promote myeloid-derived suppressor cell differentiation. So taken together, we hypothesized that CAF may facilitate the recruitment of monocytes to local tumor microenvironment through chemokines. That's why we see a positive correlation between CAFs and monocytic myeloid cells. And CAF can further affect monocyte differentiation locally to produce an immunosuppressive phenotype. To test our hypothesis, we first examined if conditioned media collected from the tumor-derived CAFs could induce monocyte chemotaxis in a transwell migration assay. So compared to control, you can clearly see that the CAF conditioned medium can increase monocytes migration. We also want to tease out which chemokines specifically mediate this enhanced cell migration. Individual recombinant human proteins were added to the medium at concentration detected in the conditioned medium and measured for their ability to attract monocytes. We found that CCL2 significantly increased monocyte migration compared to other chemokines. We next examined the abundance of chemokine receptor expressed on monocytes, and indeed we found that more than 90% of monocytes express CCL2 receptor, CCR2. We then investigated the impact of CAFS on monocyte differentiation. So we co-cultured CAFS with CD14 monocytes isolated from peripheral blood to compare the phenotype of CAF-educated cells with different monocytic myeloid cell populations. We differentiate monocytes into M1 or M2 macrophages, immature dendritic cells, as well as myeloid-derived suppressor cells using previously established cytokine stimulation methods. Compared to dendritic cells and macrophages, so you can clearly see that CAF-educated cells have reduced DC markers as well as reduced HLA-DR as well as co-stimulatory molecule CD86 expression. And those CAF-educated cells phenotypically resemble the myeloid-derived suppressor cells. So one of the gold standard of myeloid-derived suppressor cells is to inhibit T cell migration. So indeed, we found that those CAF-educated cells can robustly inhibit autologous T cell proliferation stimulated by CD3 and CD28B. So we can clearly see that from our previous results that CAF can promote chemotactic migration of monocytes and alter monocyte differentiation into myeloid-derived suppressor cells through chemokines and cytokines. We decided to use two ways to intervene this malicious interaction. So the first one is we can block the recruitment process. So we are able to find a commercially available CCR2 inhibitor to block the CD14 chemotaxis migration. And indeed, we found that blocking CCR2 can inhibit some monocytes migration. So other way we can target this CAF monocyte interaction is that we can inhibit the immunosuppressive molecules expressed by MDSCs. So one of the targets we're interested here is IDO1. And we're able to we throw in an IDO inhibitor in this T cell myeloid cell co-culture system. We can see that the IDO inhibitor can only partially reverse the immunosuppressive function of myeloid cells on the CD8 T cell proliferation. To explore additional targets mediating CAF-induced immunosuppression, we are likely to collaborate with the chemical biology department in our company, and they help us to perform proteomic analysis on MDSCs and versus the non-immunosuppressive immune population in mature dendritic cells. And we are able to find several differentially expressed proteins in MDSCs versus IDCs. And using those differentially expressed proteins, we're able to run gene ontology analysis. And one of the pathways that drew our attention is oxidative stress and activation of NADPH oxidase. So the enhanced production of reactive oxygen species has been well documented to impair T cell proliferation and activation. And interestingly, we found that in the highly expressed proteins in MDSCs, there are several components that belong to one of the NADPH oxidase family member, NADPH2, that is also known as NOX2. So this complex have several components. So one is CYBA, which is upregulated in the MDSC by the proteomic analysis. CYBB is one of the major components, is also upregulated. And we want to further test whether the function, whether DOX2 is one of the effector molecule to exert the immunosuppressive impact. We're able to find a selective NOX2 inhibitor. And by throwing this inhibitor into this T cell and myeloid cell co-culture system, we found that this inhibitor can reverse the T cell, the myeloid cell suppressive function on T cell in a dose-dependent manner. Consistent with the reversal effect, we also found that the addition of this inhibitor can reduce the superoxide production in the CAF-educated myeloid cells. With that, I'd like to summarize what I've presented so far. We think the CAFs in the tumor microenvironment can secrete chemokines such as CCL2 to recruit the CD14 positive and CCR2 positive monocytes to the tumor microenvironment. And those CAFs can also secrete cytokines or other soluble factors to further differentiate the monocytes into myeloid-derived suppressor cells, and those cells can further produce immunosuppressive molecules such as IDO1 and NUX2 to dampen the T cell response in the tumor microenvironment. With that, I'd like to thank my colleagues at Merck. So first, my postdoc mentor, Phil, who now is the Vice President of Bicycle, and also like to thank my current manager, Lily, who is super supportive. And I also like to thank my collaborators, Carlo and Josephine, for supporting this project. And also I want to take my time to thank our site-off core facility to help us fine-tune the instrument and help us to run some of the slides. And also I want to thank our site-off and IMC super user at Merck, Doug, who give me a lot of instructions and help on what are the right antibodies to test and what's the protocol for IMC. With that, I'd like to take any questions.